Good morning, everyone. Yes, so today I will tell you about how we're using computers and computation to better understand life. But I think we will start first by saying, why is it at all interesting to study biology? And in my opinion, I find this area fascinating because this is the study of life. This is where we study how we operate, how we evolve, and how do we get disease, and all the organisms and microorganisms that are operating in such harmony around us are working. And everything is the basic elements of every living system are cells. So how do they operate in such a coordinated manner? And all the plants that are surrounding us, and genes. So you all know that about 60 years ago, the structure of the DNA was discovered, and here we are 60 years later in the middle of the genomic revolution with a decade ago where we sequenced the human genome, which shed more lights on heredity and lots of more understanding in terms of medicine and so forth. So this is something that is truly interesting and fascinating. And I will start by giving you some glimpse into the complexity that is hidden in this kind of study, looking into cells. So in this movie here, can I get a bit of sound, please? Shows the amount of cells that we have in our body, the amount of types of cells with different sizes and different shapes. Okay, so now that you see how complex we are, one of the most interesting question and most fundamental question in studying life is the question of specialization. So we have cells that develop in a certain way, and the question is, how do we evolve from one single cell, and we have all this variety of different types of cells, like muscle cells and blood cells and nerve cells. This is a very critical and fundamental question, And the problem is that when the decisions are taking place, they have to be taken in a very controlled and precise manner, because when we have cells taking wrong decisions, that's how we get disease. So these kind of questions that we need to look into and study are very critical. So when we get cells take the wrong decision, that's how we get, for example, 6 fingers instead of 5 fingers, because at the course of development, one cell or a group of cells took the wrong path. So this is something that is very important to study. Now, you obviously appreciate that these kinds of systems are very, very complex and very complicated in terms of the amount of types and variety of different lineages that we need to create. So we need to somehow bypass this kind of complexity that is hidden in every single one of us in order to study these systems. And one way to do that is that it takes about 12 years to reach these amazing machines like you are today. But we can study simpler organisms like worms, for example, which develops from a fertilized egg into a hatched adult worm in the course of 72 hours. And then there, everything is much more simple. So what you can see here is an example of such an organism that we study, and it shares many characteristics with human biology. So we can study cancer, we can study aging and other kind of disease, but in very small and simple a creature like this worm. This is an example of a creature that is about 1 millimeter long. It is transparent, so we can actually see the entire development under the microscope, and it has only about 1,000 cells compared to the amount of cells that we've seen that we have. So this is very, very simple, and this is one way that we can bypass that kind of complexity that we need to tackle when we're trying to understand how we work. So, so far, we haven't said anything about computers, because we know today how to engineer and create programs of these very complicated machines like a fighter jet or a modern concept. In those kind of systems, we have many computers that are operating. So in the fighter jet, we have thousands of computers and many programs that are running behind those computers, and the same hundreds of computers that are running on these cars. So the idea is that we can use the same kind of techniques that we're using to create the programs that are executing all these computers in this engineered system, but rather than use them for this engineering, we can use them also to reverse engineer how a cell actually works, and then we can create these programs to program cells and also to simulate them and follow their behavior and try and understand them better. So that's where the computers and computation come into place. Now, normally what you would see is a computer program that looks something like that, where we have these lines and lines of codes, and these are basically instructions for the processor to create a certain behavior and to satisfy a certain program. But obviously, if you imagine that each and every process that we're trying to mimic now is a cell or a protein or a gene, and you've seen that we have so many of those and we have to have them operate in parallel, we can't use these kinds of codes and that is expressed in the form of text. So we devised other means, other languages that are more visual, that help us deal with that kind of complexity in the case of complicated systems like fighter jets and car, but we can also harness that for the sake of complexity that we observe in cells and in biology. And what you can see here, this is a visual language where instead of writing everything in text and in lines of code, we can describe the states of the different components of the system. Again, this can be cells, these can be other entities in the biological system. We describe the states and how they change from one state to another state due to some event that happened in the system. And this is a much more comprehensive and much more intuitive way to look on these kind of complex systems. Now, again, we are still tackling a problem that when we have a lot of these and we want to simulate them, we need to follow the behavior, and we can't do that in a simple manner. So what we can do is the processor can highlight exactly, like you see here in pink, where we are in the program of a particular cell. And this is, for example, an example of a small program of a part of a cell that is operating, and each one of the rectangles here represent another component inside the cell. It can be a gene, it can be a signal that is changing from one state to another state due to some other signal that is operating in the cell. And this is something that is much easier to look into and to try and understand what is going on. Now, we can handle these kinds of programs that are visual and friendly, and we can use them to a larger scale. But as you can see here, we have something like 6 components, and every time something else is being lit up and highlighted, and we know where we are in the system. But this is something that can work for this about this size of component. But imagine what happens if we have a system which has hundreds or thousands of cells. Again, this won't scale. So then we move to something else that is called reactive animation. What we have is an engine that basically connects to the program that is hidden in the back end. So you don't see any line of code here, you don't see any program. What you see here is a reflection that is fed directly from the program that represents a certain biological behavior. And this animation, as opposed to the animation that I've shown you before, which was designed by, the movie that we've seen at the beginning was designed by a person, and the person decided which cell will be progressing and in what way. Here, there's no decisions that are taking place by any person. Everything is driven directly from the model. So it's just a reflection of the model, and we can really follow and we can see what exactly are the cells doing, how are they differentiating, how are they making the decision to become of a certain type versus another type. Different types of cells are designated here in different colors, and they migrate along a certain organ. So in some cases, 2D is not enough because we're talking about the development of a structure of an organ. In this case, this is a model of the pancreas. And we have a 3D structure. So we also want to visualize the fact that cells are responding to the microenvironment, to the close environment around them, and they respond to signals which directs their budding, direct their direction of movement, direction of development, direction of division. And you see here that this is, here you have a picture of the real organ, and you see that the animation is very much like that. So we can use the same kind of animation also in the case of a 3D, but still there are quite a lot of challenges in respect that even with these kinds of animation that are showing us, and we can follow them and we can follow a certain biological process that we're studying, we still want to ask more powerful analysis questions that we cannot address just by using these kinds of simulation and animation. And the second thing is that we really want all these kind of programs to be done by the specialist, which is a biologist in this case, and not just any programmer. So in this example here, you can see again, so once again, you can see here an animation of a process where cells are dividing and migrating along a certain tube, which is part of an organ of the worm that I've shown you earlier. Now, again, this is very informative, and this in particular is an animation of a process, as it's called programmed cell death, because cells are at some point need to decide to die, and we really want to understand what are the mechanisms that lead to this kind of cell death. So in this model, we mimic all sorts of chemicals that surround these cells and how they migrate and they differentiate, and eventually they get out of this tube, and and there is a certain area here where they die, and we can stop the simulation and we can check what were the concentrations and how did they die and why did they die. But even with that, we we have still these two problems, that we can't ask questions that are beyond the simulation, beyond the visualization that we see here. Things like querying the system with specific conditions that lead to specific execution, specific endpoints of the system. This is one thing. And the second thing is that to create such a program and such an animation, you really need a professional programmer, and this is not something that can be done by every biologist. So what we also need to provide, these are tools that are almost doing these kinds of things almost automatically by dragging and dropping these elements that the biologists are interested in modeling. In this case, we have a cell, and these are proteins, and they are dragged, and this is a receptor that sits on the membrane and receives signals from other cells. And this is the kind of canvas that we can draw these kinds of elements into. And eventually, what we can also do is get the other type of analysis that I mentioned that we're still lacking, where we can query the system. We can ask about specific conditions that are happening, and we get a picture telling us whether this was satisfied or not according to this particular program. So to wrap up, I think, like Andrew said before, and also Thor, computers are all around us. We are surrounded by computers, and they affect every day of our life. And I think we're in the middle of this computer revolution that will definitely change the way we think about, and the way we think, the way we do everything. I think it's amazingly challenging, because we're now trying to use computers to fight disease, to understand the very basic principles of life, to understand things like development. And in order to do that, we really have to provide the means, the programs, and the tools to allow us to get such a better understanding. And these are quite amazing challenges, and this is exactly the kind of things that we do in Microsoft. So thank you.